Thanks very much. And first, I like to find the pointer. Good. Yeah, I'd like to thank the organizer for giving the opportunity to be presenting here our work on large-scale models of signaling networks from phosphoproteomic data. So, how do I move the slides? Yeah, so allow me a little introduction for those of you less familiar with the proteomics field. So proteomics is the omics of choice if you can do it to study signal transduction, because proteins are the main players of signaling. And within proteomics, there are a number of technologies available. Many of them are based on antibodies, and these are powerful technologies that allow you to measure many different proteins under many conditions, but they're limited by the fact that you need to have a good antibody to measure the protein of interest. And in contrast, mass spectrometry technologies allow you to measure many more proteins. In principle, go up to thousands of proteins because you're not restricted to having an antibody. But this comes at the price of being able to do much fewer experiments, because it's a much more intensive technology to do. So then, besides the issue of the number of experiments that one can do using mass spectrometry phosphoproteomics, there are a number of other challenges. One of the challenges is that in mass spectrometry, you sample the proteome in a more or less stochastic manner, and thereby, if you do twice the same experiments, you don't necessarily are able to measure the same proteins. And this list of issues, if, for example, you want to compare different studies that you don't have the same measurements and you have missing data, and also it's a relatively noisy technology, and therefore reproducibility is limited. And if you want to measure besides protein also the activity of proteins, often what you measure is modifications of these proteins, and in particular phosphorylation that we know often alters the functioning of a given protein. So if you go this path, which you can do also with antibodies or with mass spectrometry technology, you have the added complication when you use mass spectrometry that, again, you are measuring many different phosphorylation sites for which you don't really know what's the functional implication for the status of a given protein. And due to this complexity and challenges that I just described, it's really hard to build dynamic models of the signaling mechanisms, and in particular of how a cell responds to stimulation or perturbation at the signaling level. And so coming back to the other technologies, if you look at methods that you can apply for antibody-based platforms, there are many of them. So you can write the biochemical reactions, convert them in differential equations. You can look at signaling networks as logic circuits. You can infer with or without prior information using Bayesian networks and so forth. But in contrast, there are really not so many options if you want to model in this way data obtained with mass spectrometry phosphoproteomic. Of course, there are many powerful computational methods to analyze this data. You can apply different statistical tools, clustering, enrichments, and so forth. You can map this data into protein networks, but if really you want to model the dynamic functioning of signaling networks and how they respond to perturbations, there is really not many methods out there, and that's what we try to address in this study, to develop a method to build dynamic models from phosphoproteomic data obtained with mass spectrometry. So in our group, we quite like logic models because they are a relatively simple formalism that allow you to encode some of the underlying mechanisms of signal transduction without much complexity, and therefore you can look at large systems. So then we thought that maybe this is a useful platform to use for this question. So over the years, we developed an approach to build logic models for signal transduction, but for the other type of proteomics, for antibody-based proteomics. And the idea is quite simple. So you would build some signaling network based on either your own expertise or from different databases. Then you will perform experiments where you stimulate or perturb a cell with different ligands or drugs. So for example, in this toy example here, if we are interested in the pathways downstream of EGF receptor and TNF receptor, I would stimulate with EGF and TNF alpha and perhaps block the activity of a kinase. And then after this stimulation at a certain time or at multiple times, I will measure the activity of some of the components of these pathways. Again, as I said, using typically antibody-based methods. And then I will see how these components evolve over time. And then what we do is to map this data, this activity data, typically phosphorylation data of specific kinases on the networks, and we effectively train the network to the data so as to obtain the logic model that is based on the starting network and has a specific combination of logic gates that best explain the data at hand. And we develop a tool which is free open source called CellNode for doing this. So this works quite fine with antibody data. Now the question is, can we try to apply these ideas in the context of proteomic data, taking into account all the challenges I described before? So the first challenge that you have is that you cannot just go and rely on your standard pathway knowledge. Because as I said before, when you do a mass spectrometry experiment, you measure many phosphorylation sites for which you don't know which effect they have on the functioning of the protein. So you cannot just go and do this mapping of saying, okay, if I measure the activity, the phosphorylation of RAF1, I can map it on the protein RAF on my pathway. So instead of this, what we decided to use as a scaffold of the protein network is a collection of information about which kinase phosphorylates which proteins, and also which phosphatase dephosphorylates which proteins. So this information about relationships between kinase phosphatases and their underlying substrates, which are phosphorylation sites. And from this information, we build a large network. Then, if we have phosphoproteomic data that obtained under different stimulatory conditions, then we take this data from the experiments and we just do some simple statistical transformation to assess if the phosphorylation site is perturbed or not perturbed. And once we have the data normalized and we have our network, what we do is an optimization in the essence, in the same spirit of what I showed you before for antibody-based methods, where we sample from this huge network of kinase and substrate relationships, we sample sub-networks. For all of them, we simulate the conditions for which we have experimental data, and we do it as a Boolean model, as a very simple logical circuit which proteins can be on and off. And then we compare these simulations with that we obtain from the data, which is also on and off values. And we do this in an iterative process where we try to sample the space of potential networks, and eventually this algorithm converges into a set of networks and of frequency of edges in the network. So what this process provides us then at the end of the day is a Boolean logic model of a large kinase substrate network that provide us the most likely paths that connect the perturbations, either ligands activating pathways or drugs blocking specific kinases, and all the phosphosites on the proteins downstream of these perturbations in the cell. And these links are functional, are causal, in that they tell us how you go from a perturbation down to the change in the phosphorylation site. They are context specific in the sense that you have obtained them by adjusting them to experimental data obtained in a specific context. And we have also shown that they are predictive of what's going to happen in new nodes or in new sites. And all this approach is implemented in a free open source tool that we call PhoNeMs, which stands for Phosphorylation Network for Mass Spectrometry. So as a case study to illustrate this approach, we work on data from our collaborator Pedro Cutillas in Queen's Mary Hospital in London, and his student Ed Wilkes. What he did is to take MCF7 breast cancer cell lines and perturb them with 20 different inhibitors. So these 20 inhibitors affect 10 different kinases, so 2 inhibitors per kinase, with the idea that by using 2 different inhibitors, we can tease out some of the off-target effects, because no kinase inhibitor is perfect, and this way we wanted to control a bit these off-target effects. So then what I did is to take these samples, put one drug, wait for an hour, and then measure with shotgun mass spectrometry the activation of around 12,000 phosphosites for each of these conditions. And then what we did is to take this data and run it through the pipeline I just described and obtain then a very large network. This is even only a small part of it, which covers all of these changes in the phosphoproteome under all of these different conditions. And now the question is this useful in the sense of learning new biology? And for this, what we did is to go into the model and to try to look at the different pathways that were predicted from the model and seeing if there were some interesting biology. And so as one example, this approach predicts that there is a pathway linking mTOR to CDK2 and some of its targets, like cyclin D1, via AKT. Right? So this is just from taking these snapshots of phosphoproteomic data and kind of passing them through these network diagrams. But if this is truly a functional link from the perturbation here all the way down here, it should be that if now you do another experiment where you block mTOR, but also if you block the intermediate node AKT, you should see a functional effect which you would expect by blocking the activity of CDK2, which in this case would be primarily that cells should be unable to go in the cell cycle from the G1 to S phase and should be arrested in G1 phase. And when we did this experiment, we found exactly that. So this is untreated cells. These are cells treated with 2 AKT inhibitors and 2 mTOR inhibitors. And as you can see, they get stuck into G1 phase, and they stay there, and they don't move on to the next phase of the cell cycle. So this is an example of how you can take these huge data sets and filter them through these kinase asteroid networks and obtain fairly specific pathways and hypotheses, and then you can follow up experimentally. The second use that we found we could try to do is to look at this problem I told you at the beginning. If you run 2 experiments under the same conditions, given the stochastic nature of mass spectrometry, you are unlikely to see the same phosphorylation sites, and therefore it's really hard to integrate or to compare 2 experiments. But we thought if you filter this data through these networks, you may be able to find kind of the robust pathways of activation that should be indeed consistent. And to answer this, we took the same data set I showed you before, but also a second data set from the same group. This is a data set that I had generated 1 year before, because they were in Imperial College in London. They moved to another university. So it was the same group, but different mass spec machine, different environment, and also slightly different set of inhibitors, and actually a different coverage of phosphorylation sites. So the data itself doesn't overlap much, but if you run both of the data sets, what you find is that there is a good consistency on the underlying pathways that you're able to identify. So as an example, if you look at what is downstream of mTOR, even though the number of the specific phosphorylation sites between the 2 studies, you will see a very little overlap, only 2 of around 20. The number of kinases that you identify in the reconstructed pathways is around 70%. So you're able really to extract much more robust insights about the underlying functioning. And with that, I'd like to finish by first thanking Camille, who was actually my first PhD student. He was a great student. He did all this work in collaboration with Ed, Pedro, and Pedro in London. And I leave you with the key message of this talk, which means that these method phonemes enable us to combine mass spec phosphoproteomic data with kinase asteroid interaction information from databases to construct logic models that can provide us robust results across different data sets, and also can provide us testable hypotheses, as I showed you in the example 2 slides ago. With that, I'd like to thank you for your attention, and I'm happy to take any questions.
Thank you, Julio. You took the data at 1 hour after stimulation. So what will happen if you measure at different times? Would you recover slightly different network, or can you infer things that you wouldn't infer at 1 hour?
Yeah, so I would expect to see differences, and of course, signal transduction is a very dynamic process, so it's waves of activation, and taking a single snapshot is a limitation, but you have to balance time points with perturbations. So you could do, I mean, we haven't done that in this context because we have only 1 time point, but when we do the logic models of antibody-based data, you can basically expand the approach to model different snapshots as almost like a Markov-Chain time of process, if you like. So there are ways to even use different, basically time series data to analyze it as a whole, which is even much more informative. But for sure, you will see big differences.
On that last point about reconciling multiple data sets, can you actually get further insights into that in terms of why you get distinct ones that are activated? Is there something to do with localization or scaffolding, or what is it about those proteins in one cell type versus another that are under the same mTOR control but different things happen?
So,
or did I not get that right?
So that the kinase that you identify that they are not the same, even though they are more consistent than the raw data. So we didn't look into much detail. I mean, I think it's because still the data sets are quite different, so they don't capture, even the coverage was 8,000 versus 12,000 phosphosites. So I think it's because even though the method allows you to filter to some degree the information, still the data sets were quite different. It could be that there are some specific biology on some of these proteins or some of rather their substrates that affects how they run through the mass spec and therefore affects how robust they are. So it is known like high abundance proteins, for example, are much more consistently reported in different experiments. And maybe it has also to do with that, but we didn't look into detail. But it's a good question. Oh, I now have a mass spec expert. Uh-oh.
I was just wondering what quantitative technique you used for the mass spec.
So, I mean, it's label-free, relatively standard shotgun proteomics, but in the lab of Pedro, they have their own in-house analytics, they call it Pascal, that they build that. It, compared to the data set I've seen, is quite good for label-free. And another good thing is that we had both biological and technical replicates, so we could assess relatively well significance of the values. Yeah.
Okay, and then you, as far as the input to your model, it's on or off phosphosites?
Yeah, I mean, it's a compromise that we have to make because, I mean, what you do is you assign each phosphosite in a given experiment, you assign this to this Gaussian distribution, so you more or less say it's more likely to be on than off. But for the modeling purposes, you have to, you simplify to being on and off, which of course is not true in biology, but it's a way to be able to handle all the data together in a model.
Thank you very much.
Thanks.
Quick question, if I understood the initial part when you were motivating it, so you think that logical models are better suited for this kind of data, or is this just something that...
I mean, better...
I'm not anti-logical models, I'm actually pro, but I'm just...
Yeah, I mean, better than as opposed to what would be the other methods. You mean like ODEs or...
The Bayesian method.
Bayesian. I mean, so Boolean models, we like them because they're very simple. It's almost as simple as they can be, but you can, and thereby you can look at large networks, large systems, large amount of data in an efficient way, but they still allow you to encode in these networks the knowledge that you have about the systems. So we haven't explored other related approaches in the context of the mass spec, but for example, for the antibody-based methods, you can then move from the Boolean to an ODE model. You can also, we have work where we combine Boolean models with more Bayesian or data-driven methods. So all methods are have pros and cons, and in our hands, Boolean model is a good first step in a specific data type because they are so simple that you can really understand what's happening.
Okay, thank you.